much of our work has been looking at the microbiome in atopic dermatitis patients. And some of the interesting things that we have found are based on strain level differences. And it highlights how fairly individual one's microbiome is. We see that for healthy individuals, it is fairly individualized, that the microbiome we see in a healthy individual can be very different from one person to another. And similarly, in atopic dermatitis, we see a particular strain of Staphylococcus aureus that seems to be highly predominant during flares of the disease in patients who have severe atopic dermatitis. So in one of our studies, we looked at those isolates that we collected from those patients, and we tested it in a mouse model, and we're able to show that there's strain level differences in the biological response in mice and mouse skin, suggesting that there might be a role for these particular strains of Staphylococcus aureus in atopic dermatitis. But it's fairly complex, and that's some of what our session discusses, is that there's the immune system that's also very important, barrier is important, and talking about different therapies, having the discussion about what is the role of dilute bleach baths. We know that those can be helpful in certain situations, particularly in maintenance of atopic dermatitis, but also the complexities of all of the other aspects of environment as well in the disease and genetics. So atopic dermatitis is fairly complex, and there's still a lot more research to be done. So there is a lot of work that's been looking at Staph aureus and the skin barrier, and how in some studies they have shown that Staph aureus may play a role in breaking down the skin barrier, and in other situations where a weaker skin barrier may actually allow Staph aureus to connect with the host or interact with the host in a different way. And so teasing that apart and trying to figure out who is doing what is complicated. Ideally, we would be able to study this in humans, but that is not that feasible. And so we have to extrapolate and look at mouse models or even in vitro studies so that we can try to understand and elucidate the different mechanisms. But it's not completely understood at this point. So how could we use the microbiome to treat diseases? I think the best examples of that are primarily in the gut, and where people have had very good success in using fecal microbiota transplants to treat something like Clostridium difficile colitis. The challenges there is that is an infection, whereas a lot of the skin disease that have been studied are inflammatory conditions. So I think it's going to be a lot more complicated, and trying to figure out what role does manipulating the microbiome have in some of these inflammatory multifactorial diseases. And so based on the studies that have been done, for example, probiotics, we have mixed results. In various different studies, there have been some studies that have looked at trying to manipulate the skin microbes. And so there may be an option for that as well, but given that it's a multifactorial disease, it would be difficult to say, here is one product and it will treat everybody equally well. So what are the exciting questions that still have to be elucidated, I think, are what is it that microbes are doing, particularly bacteria, in atopic dermatitis? I don't think we have a full understanding of that. But on the other hand, with these primary immunodeficiency syndromes where you have a specific genetic defect and it changes one's microbiome, that actually is giving us a lot of information of how specifically our immune system is shaping things. And so trying to figure out that potentially, while those are rare disorders, they may actually give us insights into a more complex disorder.